If you look at corporate sustainability reports today, global green biomass appears to be rising steadily, yet true ecosystem health continues to collapse. This happens because modern conservation relies on an accounting system that treats living, dynamic organisms like static inventory in a warehouse. We count biological mass using visual taxonomy, recording the surface-level physical traits of plants and animals. We measure their shape, size, and leaf structure, rather than auditing their actual genetic integrity. Relying solely on these visual headcounts creates a dangerous illusion of biodiversity. Grouping genetically distinct populations under a single blanket classification because they look similar, we mask localized, silent extinctions right in front of our eyes. Without a genetic audit to verify these physical headcounts, we are left with a biological ledger that lacks the resolution to differentiate between real growth and a statistical illusion. Traditional taxonomy was built in the 18th century. It relied on naming things based entirely on their physical appearance. That works in a static environment, but it fails completely in complex, evolving ecosystems. This outdated approach creates what biologists call taxonomic noise, severe data corruption in our environmental ledgers, caused by duplicative naming or false groupings. Nature routinely uses convergent evolution to survive. Unrelated organisms adapt to look identical simply because they face the exact same harsh climate conditions. Prioritizing visual shape over genetic identity forces a trade-off. Conservation resources are directed toward protecting environmental phantoms, while truly distinct evolutionary lineages remain invisible to the law. To see how visual taxonomy actively fabricates data, we have to look at phenotypic plasticity. This is a biological mechanism where environmental factors like temperature, soil chemistry, or shade alter an organism's physical traits without changing its DNA. Consider the arrowhead plant. Depending on water depth, its single genetic identity expresses 3 completely different leaf shapes: underwater ribbons, floating round leaves, or arrow-shaped stalks. Traditional surveys log these as 3 separate species, artificially bloating biodiversity counts and creating a statistical surplus. Now look at the exact opposite problem: apomixis, or self-cloning, in plants like the dandelion or the blackberry. These plants frequently drop seeds that are exact genetic clones of the parent. As these clones spread, minor micromutations in their leaf hairs lead taxonomists to name hundreds of phantom microspecies that possess no unique evolutionary value. This explosion of cloning noise clogs international registries, completely paralyzing policymakers trying to identify which wild plant lineages are actually endangered. Because plants freely morph their shapes and clone themselves, a raw head count without a strict genetic verification protocol is nothing more than a statistical mirage. Beyond natural taxonomic confusion, we face the anthropogenic blur. The unrestricted expansion of agricultural cultivars, engineered crops, and lab biotypes has completely shattered the boundaries of natural evolution. This triggers genomic swamping. This occurs when massive populations of human-engineered crops interbreed with wild relatives, silently erasing the unique wild genome. Take the boundary between wild rice and domestic rice. When commercial farms operate near natural wetlands, billions of wind-blown agricultural pollen grains overwrite the deeply evolved, climate-resilient genes of the wild rice. A surface-level ecological surveyor will report a thriving, heavily populated green wetland. But genetically, that unique wild species has gone extinct, entirely replaced by an agricultural product. This applies to aquatic environments as well. Every year, hundreds of thousands of selectively bred Atlantic salmon escape from commercial aquaculture pens directly into wild waterways, breeding with fragile native populations. Surface headcounts of these rivers show massive numbers of fish. Policymakers read these inflated ledgers and celebrate conservation victories, blind to the genomic pollution happening beneath the water. When human-engineered variants escape into wild zones, they don't just add to the biodiversity count, they actively delete wild ecosystems while falsely keeping the global biodiversity count high. Massive global corporations use these flawed taxonomic ledgers for ESG reporting, specifically through carbon offset tree planting. They exploit a glaring accounting loophole. Current international frameworks treat one tree stock as one unit of biodiversity data. The ledger entirely ignores whether that tree is a wild native species or a sterile, mass-produced clone. A true natural forest operates as a syngonion, a complex network of diverse native species sharing nutrients via underground pathways, maintaining deep genetic variation. Commercial palm and eucalyptus plantations are the exact opposite. They are genetically uniform, non-native clones, engineered solely for rapid wood density. They offer zero ecological value to native wildlife. Furthermore, these monocultures frequently exhibit allelopathy. They secrete compounds that chemically starve the soil, actively preventing any native understory plants from surviving in their shadow. Yet corporations register these sterile dead zones as reforested ecosystems in international climate accords to secure green tax credits. 10 million cloned industrial units planted in a grid are a crop, not a forest. Raw biomass must never be conflated with true biodiversity. To stop this data corruption, we must legally and mathematically demarcate natural wild flora from anthropogenic industrial crops. This requires the implementation of a bio-audit. By deploying environmental DNA sampling and high-throughput sequencing in the field, we can instantly filter out taxonomic noise and identify sterile clones. Global tracking requires a strict binary partition. On one side, Category A, natural wild populations, captures naturally evolved organisms with genetic integrity. The absolute legal rule of this framework is that Category B mass must never be aggregated into conservation data to cushion or offset the decline of Category A wildlife. We must replace absolute headcounts with effective population size, a metric that measures only the genetically unique, breeding-capable individuals, discarding sterile clones from the data pool entirely. By shifting from morphological naming to strict genetic accounting, the bio-audit permanently closes the greenwashing loophole. The bio-audit standard is useless if it remains contained within academic laboratories. It must be integrated directly into global financial and legal frameworks. International bodies, like the United Nations Environment Program, must formally recognize and enforce the demarcation between Category A wild biomass and Category B engineered biomass. Institutions managing the IUCN Red List must restructure their metrics, officially screening for genomic swamping and cryptic extinctions before issuing global conservation statuses. This culminates in audit-gated financing. Sovereign governments and global banks must freeze all climate and conservation funding that relies purely on visual checklists and unverified ESG reports. Global capital must only be unlocked for projects that can prove their genetic baselines through third-party bio-audits. The international effort to stabilize the biosphere is being managed through a ledger that fails to distinguish between natural wild populations and human-engineered crops. Human engineering and clonal agriculture will continue to mask silent, localized extinctions as long as we only measure surface greenness. True nature conservation demands that we draw a hard, legally verifiable line between the hand of natural evolution and the footprint of biotechnology. The scientific and policy communities must abandon unverified guesswork and adopt precise biological accounting. We cannot protect what we do not accurately measure. The survival of global biodiversity depends entirely on a clean ledger.